Hi there. This is part 2 of organelles, so I'll cover primarily um, chloroplasts or plastids, and then cover a few other eukaryotic organelles, and then talk a bit about prokaryotes as well at the end. Here's the general form of a plastid, or chloroplast in this case. It has an outer membrane, and then an inner membrane here, and an inner membrane space between them. And then there's an infolding of the inner membrane to form these thylakoid membranes. The cytosolic part, or the part that would be a bacterial cytosol, is called the stroma. And then inside of each of the thylakoids, there's a lumen that become acidified during photosynthesis, during the light reactions of photosynthesis. So the first chloroplasts were cyanobacteria that were engulfed by protists, a eukaryotic cell. And the cyanobacteria by that time were oxygenic, so you have even more oxygen being given off after you get the first plants, which were glaucophytes and red algae, and then later on green algae. These chloroplasts or cyanobacteria originated back here when you got oxygenic photosynthesis. Cyanobacteria themselves date back to probably around 3.5 billion or more years ago, but at that time they were not oxygenic until they got the manganese center here about 2.7 billion years ago and became oxygenic. And by that time they had photosystem 1 and photosystem 2, and then also had the Calvin cycle for fixing carbon dioxide into organic compounds as well. So then on a phylogenetic tree, again, this is the root back here about 3.8 to 4 billion years ago, bacteria over here, including the cyanobacteria, and the cyanobacterium, one of them, became the incorporated into a protist cell, a eukaryotic cell, and that became the archaeplastida, or the plants. So in our conceptual scheme here, here's a cyanobacterium that already had photosystem 1 and photosystem 2, as well as the Calvin cycle, was taken up inside of a eukaryote that had a nucleus and a mitochondrion by that time, but was not photosynthetic. So you can see here, this is the first plant cell, probably something that looked like a glaucophyte or red alga about 1.9 billion years ago. So these were the initial photosynthetic eukaryotes. Now since then, there have been several other eukaryotic photosynthetic organisms that have evolved, and we'll talk about those near the end of this video. Here are some different views of photosynthetic tissues and chloroplasts. So this would be a cross-section through a leaf where you'd have what are called palisade cells or palisade parenchyma that would have chloroplasts around the edges, and this would be on the upper surface of the leaf, and then you'd have the lower surface of the leaf down here, the lower half of the leaf, containing spongy parenchyma with chloroplasts in them, and a lot of gaps between, and that's mainly for gas exchange to take in carbon dioxide and to release oxygen. And then on the bottom of the leaf, there are cells that can open and close that region, and those are called stomata. And the cells that open and close are called guard cells down there. So that's just a general picture of a leaf. And then down here on the bottom, you can see a chloroplast in some of that tissue. And this is a scanning EM, so you can see the surfaces. And if you look really closely on these thylakoid membranes, these stacked regions, you can see little balls on the end, and those are the ATP synthases pointing into the stroma. So you see a general picture over here, and up here's an EM, a transmission picture of a section of a chloroplast, and you can see the grana, the thylakoid membranes stacked in what are called grana. So they're usually oriented so they get exposed to a lot of sunlight to do the light reactions of photosynthesis. And then the other sections of the chloroplast are the stroma, which is sort of the cytosolic part of what once was a bacterium. And then you can kind of see the outer and inner membranes. Here's an artist's depiction of those. So you have the outer membrane, which is more or less surrounds the whole organelle, and then the inner membrane, which also surrounds the inner part of the stroma, and then the thylakoid membranes are inside of that do the light reactions, and the dark reactions, the Calvin cycle, takes place in the stroma. So within those organelles, you have light harvesting complexes that energize chlorophyll molecules using light energy. And then the energy is transferred to photosystem 2, which moves electrons and in the process produces oxygen from water, so splitting off hydrogen, oxygen, and electrons. The electrons then are moved through a series of cytochromes and other proteins to photosystem 1. And then finally, the electrons are transferred through several ferredoxins and added to NADP, with a proton to form NADPH. Through this process, too, protons are moved from the stroma through the thylakoid membrane and into the lumen of the thylakoid. Then the ions that build up in the lumen of the thylakoid are allowed to flow through from the lumen to the stroma through the ATP synthase to form ATP from ADP and inorganic phosphate. Then in what are called the dark reactions, which is the Calvin cycle, ATP, NADPH, and carbon dioxide are used to make carbon compounds, and some of those are sucrose and starch, but several others as well. Therefore, many proteins are needed in addition to membranes, membrane proteins, ribosomal RNA, ribosomal proteins, tRNAs, DNA polymerase, RNA polymerase, etc. And also remember that plastids can develop into chloroplasts, but they can also develop into amyloplasts, idioplasts, chromoplasts, elaioplasts, and several other forms. But all of those have the same chromosomal DNA, so they're similar, but they develop in different ways based on the signals they're getting, both from the environment and from what's coming from the nucleus. Here is the diagram of the light reactions of photosynthesis. Here's photosystem 2 and photosystem 1 here, the cytochrome BF complex, and then the ATP synthase here. So the first thing that happens is light hits the photosystem 2, and then that converts water into oxygen. It strips off the electrons, which go through plastoquinone and into cytochrome BF, pumping some protons across the membrane. Here you wind up with protons also. And then through plastocyanin, and then into photosystem 1, where that electron is further energized here through other chlorophylls. So both of these have chlorophylls in them that excite those electrons. The electron winds up being added to NADP as well as a proton, so you have then NADPH, and that functions in carbon fixation in the Calvin cycle. So overall, you strip the proton off here. There are more protons move through the membrane proteins here through cytochrome. And so you build up protons in the lumen of the thylakoid. Those then flow through the ATP synthase, and using the proton motive force to actually spin the molecule, ADP plus inorganic phosphate are added together to make ATP. And again, those ATPs wind up in carbon fixation in the dark reactions, or the Calvin cycle. Here is a simple representation of the Calvin cycle, the dark reactions, also known as the reductive pentose phosphate cycle. So carbon dioxide is fixed onto ribulose 1,5-bisphosphate by the enzyme ribulose bisphosphate carboxylase oxygenase, or shortened to Rubisco. That becomes a 6-carbon sugar immediately, but is split in half into 2 molecules of 3-phosphoglycerate. Now that can go back into glycolysis or gluconeogenesis out in the cytosol, or part of this molecule can be used. It's phosphorylated. NADPH is used to reduce it, and then you further go through many steps here, which aren't listed, finally winding up at ribulose 1,5-bisphosphate again, and then through the cycle. Different things can come off of here also, including 3-phosphoglycerate, to make glucose, sucrose, fructose, starches, depending on what's needed by the plant cells at the time. So you might recognize a few of the molecules in here, 3-phosphoglycerate and glyceraldehyde 3-phosphate. So both of those are also in both the pathway for glycolysis and for gluconeogenesis. So if you go one direction, you can go into the TCA cycle eventually, or you can go the other direction and make glucose or fructose and other sugars as well. But there's 3-phosphoglycerate, and here's glyceraldehyde 3-phosphate. And remember, these are out in the cytosol, whereas the photosynthetic reactions, the Calvin cycle, is within the stroma of the chloroplast. This diagram is similar to the one that was in the last video on organelles on mitochondria, but this one's for the chloroplast. So you can see here is just a general view of a chloroplast with the outer membrane, the inner membrane space, the inner membrane, and then the thylakoid membrane, which you can see has ATP synthases all over it, and it also is an infolding of the inner membrane. And then the stroma is just the central part of that cell where the dark reactions occur, and the light reactions all occur on the thylakoid membrane and in the lumen of the thylakoid. And this diagram is primarily to look at the number of genes and gene products needed by a chloroplast to function. So the first thing you notice is there have to be proteins that are imported from the cytosol, which were transcribed and translated by the nucleus and then in the cytosol, respectively. But they have to be imported through transport channels. So those are necessary, and whether they wind up in the stroma or in the inner membrane space or in the thylakoid membrane, they will need different proteins to go to each one of those places and different transporters to carry them there. So this requires at least 10 genes for the transporters, and probably more like 10 times that amount, or probably 100 different transporters to get to the different compartments within the chloroplast. Next, if you go down here to the light reactions, photosystem 2 requires more than 30 genes to make the different protein products that come together for photosystem 2. Then there's plastoquinone, which is also more genes are needed for that. For the cytochrome BF complex, at least 10 genes are needed for that. And then there's plastocyanins, which transfer the electrons further into photosystem 1, which also requires about 30 different genes and then gene products. And then these iron proteins also are necessary one that produces NADPH from the electrons, NADP, and protons. So then you get a buildup of protons inside the lumen of the thylakoid, and those flow through the ATP synthase into the stroma, and in doing so make ATP from ADP and inorganic phosphate. And ATP synthase requires at least 8 genes, and usually it's more than that. Probably a dozen genes are necessary to produce that protein complex. Then there are transport channels necessary to bring different products into and out of the chloroplast stroma for the Calvin cycle, both input and outputs from the Calvin cycle. So at least 20 genes are necessary for that, and then at least 13 genes for the Calvin cycle itself. And here you can see more of the steps that were left out of that simpler diagram earlier. Finally, there is DNA. There's a chromosome in the chloroplasts, and they need to replicate their chromosomes. At least 30 genes are needed for DNA replication, including DNA polymerase and accessory proteins. And then at least 100 genes are needed for transcription, RNA polymerase and accessory proteins, and then at least 100 genes for construction of the ribosomes, both for the ribosomal RNA and for the ribosomal proteins. And when you add all of those up, at least 81 genes and proteins necessary for the light reactions. And I haven't even mentioned chlorophyll biosynthesis also, which is necessary in this process, and that requires more genes. At least 13 for the Calvin cycle, at least 30 for replication, and 200 for transcription and translation, and then membrane functions. So that brings the total to 374, and it's probably more than double that. So figure that number would go up to around 700 or 800 or possibly even 1,000 genes. And gene products are necessary to produce a functional chloroplast. So how many genes do chloroplasts have? Well, approximately 170 genes, but it varies from about 110 to over 200 genes, depending on the plastid. And with an average size of around 150 kilobases. And if you look at what's on those chromosomes, there are usually ribosomal RNA genes, 2 to 8 of those, depending on whether there are multiples. tRNA genes, 30 to 40. Ribosomal protein genes, 20 to 30. So total genes for translation, about 60 to 70. And we already said you need about 100 of those genes. And then transcription genes, fewer than 10. And probably more like 100 are necessary. Photosynthesis genes, 10 to 30, and multiples of that are needed. ATPase subunits, 5 to 10, so you can almost make an ATP synthase from the chloroplast genome, but not always. Often one or more subunits are expressed from the nucleus as well. And then often there are unidentified open reading frames, so it looks like a gene is there, but it's not clear what it is. And so there's up to 60 of those. It may be that some of them are pseudogenes or genes that are now, at least good versions have moved to the nucleus, so they may be expressed from there. This is the circular genome of the chloroplast DNA from spinach, and this is sort of similar to what you would find in other higher vascular plants. Normally these are arranged so you have a large single copy region, LSC, and then a small single copy region here, and then inverted repeats. And the inverted repeats most often contain the ribosomal genes within them. These yellow ones are genes for NADH dehydrogenase. The red ones are ribosomal genes. tRNA genes are in blue, so there are a lot of those. And then ones involved primarily in photosynthesis are in different colors around the ring. And the spinach genome is 151,000 base pairs in length. So with this genomic map and other maps that I'll be showing you, you can see that there are two sides on this circle. So the outer part are genes that are transcribed in one direction. on one strand, and then in the opposite strand, those are transcribed in the opposite direction and expressed in that direction. You can see this inverted repeat. The ribosomal genes are transcribed in one direction on this one, and in the opposite direction on the other strand on this the other inverted repeat. So keep that in mind when we look at other genomes as well. Here is the circular genome of a red alga, and it's about the same size as that from the plant, and it has a few more genes on it, and it only has one ribosomal copy, and that's shown in dark blue here. Otherwise, then you have a genetic system that is DNA polymerase and RNA polymerase, photosystems in green, ATP synthesis in red, metabolism in yellow, transport in purple, and unknown are gray. So you can see quite a few different categories of genes, but again, not enough to encode all of the proteins and other things needed for functional chloroplast. In order to produce a functional chloroplast, most of the genes are actually expressed from the nucleus now, rather than from the plastid genome. Also, keep in mind that in higher plants, the plastids can develop into different types of organelles. And they all have the same DNA, the same starting DNA. So you have a proplastid, which is undifferentiated. This can then develop into an idioplast. So if you have a plant that's not in sunlight, it will grow and the chloroplasts will divide, but they will not turn green. They won't develop chlorophyll. Once you hit them with light, then the idioplast or the proplastid will become a green chloroplast. In some parts of the plant, including like tomatoes and some fruits, those chloroplasts will then continue to differentiate into chromoplasts. So they'll have different types of pigment in them. They'll start green with chloroplasts and change into chromoplasts. You can also have leucoplasts develop, which are storage tissues or become storage tissues, such as proteinoplasts, storing storage proteins, elioplasts, storing fats, and amyloplasts, storing starches. And these can appear in different parts of the plant, including in the seeds, in the roots, in the tubers, etc. But again, they're developing starting with the same genome. It's really the same organism, essentially, the same organelle, but developing in different ways depending on how those genes are expressed. So looking at the evolution of this process, about 1.9 billion years ago, there was a free-living cyanobacterium with about 1,000 to 1,500 genes with a genome size of several million base pairs. And then there was a protist cell that had a nucleus and a cytosol and a genome, a large genome by that time, probably several hundred megabases of DNA and probably 10,000 genes, something like that. They probably had some sort of mutualistic relationship even before the cyanobacterium entered and became an organelle. This would allow them some time for sorting out of which proteins were compatible between the two, which genes were compatible between the two, etc. And then about 1.9 billion years ago, as I stated earlier, the cyanobacterium started becoming an endosymbiont. That is, it was inside the cell, the protist cell now. And it was a symbiont, so it was providing the photosynthate. It was giving this now plant cell, a red alga or glaucophyte, the benefit of being an autotroph now. And it was also getting things from the nucleus also, beneficial compounds that allowed it to survive there. But almost immediately it started losing genes and losing part of its genome. And this has been found with all endosymbionts and even parasites that I know of. In addition to losing part of its genome, quite a bit of its genome, now it only has 170 genes, and it's down to 150 kilobases by the time it becomes an organelle. Some of the lost copies, or all of the lost copies of the genes, actually moved into the nucleus of the host cell and became integrated into the host cell chromosomes. And in fact, if you look at the genome of Arabidopsis thaliana, about 19% of its genome is made up of cyanobacterial genes, which is about 4,500 copies of those genes. And remember, it only had 1,500 genes to begin with. So on average, it has 3 copies of each one of those genes, although probably not all of them are functional. And it's been found that some of the products from the original host cell, that protist cell, are functioning out here in the cytosol and in the membranes of that host cell. And the products from the cyanobacterial organism, the original one, are functioning by moving into that plastid to function within that plastid. But in addition to that, some of the products from the host genes, so they're transcribed here, translated, and moved into the plastid, and they function in the plastid. And some of the products from the cyanobacteria, the genes are transcribed here, translated out in the cytosol, and they function out in the cytosol. So there's been a mixing of these genomes and the genome and the products of those two genomes throughout evolution, such that chimeric pathways and cycles have evolved. So this picture shows the Calvin cycle here, and then glycolysis and gluconeogenesis over here. And all of these colored balls here are the enzymes in those reactions. So they're not the reactants and products. These are showing the enzymes. Everything in green is of cyanobacterial origin. Everything in lighter blue is actually an enzyme that originated in alpha proteobacterium, that is, from the it's been inherited from the mitochondria. And everything in the darker blue here, that's from the original host cell. So when you look at the Calvin cycle, many of the enzymes are from the original cyanobacterial ancestor. But then a number of them have come over from the mitochondria, which were the ancestors of those were the alpha proteobacteria. And then there are actually some enzymes here that are originally from the host cell, from that host eukaryotic cell. So now you have a chimeric pathway. In addition, out here in the cytosol, the enzymes for glycolysis, some of those, very few of them are from the original host cell, some are from the mitochondria originally, and then some are actually from the cyanobacterial ancestor as well. So before any of this endosymbiosis and forming of organelles occurred, each one of those organisms had a glycolysis pathway. Each of them had a gluconeogenesis pathway, and some of them had a Calvin cycle, not all of them. So there was a duplication originally of many of the genes, and through evolution, it's been sorted out. So in the case of glycolysis here, that phosphoglucokinase from the cyanobacterium worked best in the pathway. And the genes for phosphoglucokinase from the original host have disappeared, and the ones from the mitochondria, the alpha proteobacterial type, have also disappeared throughout evolution. And the tried and true glycolysis pathway now looks like this, which is chimeric. That means it has a mixture of products from the 3 different organisms in those endosymbiotic events to form the organelles. So if you look at a phylogenetic tree, and these are phylogenetic trees for several of those enzymes, I'll just go through this one, which is phosphoglucokinase. Originally, you have one gene, then you get evolution into an archaebacterial type, into a eubacterial type, and then a eukaryotic type, which comes off of this archaebacterium, archaeal clade. But it disappears. You see that one from the host disappears, and instead the one that came from the mitochondrion is here, and the one that came from the plastid is here. And in the plant, then the plastid type is used in the plants, and the other two disappear. Whereas in animals here, this is the animal ones, the one from the original host from here that came off the archaeal branch disappeared, and these mitochondrial ones from the alpha proteobacteria originally split and diversify in animals and fungi, and those are the ones that are used in glycolysis in those animal and fungi. So the phosphoglucokinase in your glycolysis pathway came from the alpha proteobacterial side and not from the original eukaryotic host. That eukaryotic host phosphoglucokinase is gone. It went extinct. So now I want to move past the chloroplasts into this group of chromalveolates, which are now called the alveolata, stromatophiles, and other related groups. Because what's happened here is you've gotten green and red algae from the plant clade here that have moved into these protist cells here to become photosynthetic. But instead of having a prokaryote inside of a eukaryote, as you do with plants, you have a eukaryotic cell, either a red or a green alga, inside of another eukaryotic cell, which was originally non-photosynthetic. So it's a eukaryote within a eukaryote. And in a few of these, you have a eukaryote inside of a eukaryote inside of a eukaryote. So there's an additional endosymbiotic event to form even more complex organelles. So it becomes quite complex. So this is a sort of a complex diagram, but to get a eukaryote here, you come off of here with a eukaryotic progenote that came off of the archaeal line. You join that with a planktomycete that has internal membranes, including something that looks like a nucleus, and now you have a eukaryote that has a nucleus. And then you have a mutualistic association between a eukaryote with a nucleus and an alpha proteobacterium that eventually becomes the organelle, which is the mitochondrion. Then you have another endosymbiotic event between a mitochondrial eukaryote and a cyanobacterium, which then become the chloroplast. And that's actually happened more than once. One that produced the plants as we know it, and then another one in the rhizaria that formed a genus called Pollinella, and that happened more recently. And there's likely another event that happened back here between two gram-positive single membrane called monoderms joining together to form the diderms or gram-negative bacteria. So that's another one. But then you have other endosymbiotic events over here between a non-photosynthetic protist eukaryote and a photosynthetic eukaryote over here, and you join those together to form a new eukaryote, which is essentially a eukaryotic cell, or which becomes the organelle inside of another eukaryotic cell. So the initial one here I'm showing is a green alga entering this protist cell, which is non-photosynthetic, to become what are known as the excavata, that group, very basal group of eukaryotes. And they form things like euglena, which is photosynthetic, but also trypanosomes, which are not photosynthetic, but do have photosynthesis genes in their genomes. But they've given up being autotrophs, and they're now parasites, so they grow on other organisms. That's how they eat. Then there are other groups down here where you get joining of either a red or a green alga with a non-photosynthetic protist to get this group, to get the ciliates, cryptomonads, haptophytes, and heteroconts. So these are those basal groups of what people loosely call algae, but the true algae are up here in the plants, right? And although these are, people call them protists or algae, they are distinct groups, very distinct groups. But after they form here with this secondary endosymbiosis, then you get other endosymbiotic events. Essentially, then you have a prokaryote inside of a eukaryote, and then a eukaryote inside of another eukaryote, and that is inside of another eukaryote. So there are many endosymbiotic events that lead to these. For some of them, for most of them, you still have a chloroplast. The mitochondria usually are not present anymore in the organelle. And sometimes a nucleomorph, a smaller nucleus, is found in some of those as well. So you can tell that there's a lot of evidence that those were eukaryotic cells before they became those endosymbionts and then organelles. Okay, so those are those. I'll show you a few pictures of some of those. But remember, these, although they look like chloroplasts, and they're essentially chloroplasts within the remnants of the original eukaryotic red or green alga. And in the case of these ones up here, they're actually, even though you can see the chloroplast and sometimes a nucleomorph, those are eukaryotes inside of eukaryotes within another eukaryote. And each one of those compartments then also carried their own genomes. So you have even a more complex mixing of all those genomes and gene products. So here are some representatives of that basal group, the excavata, including Giardia lamblia, Tracholamonas, and Euglena. And these two have chloroplasts, but those chloroplasts, right, are really green algae. So that really is a eukaryote, which is the green part in there, inside of this other eukaryote, and those are Euglena. Now, Giardia and trypanosomes are not photosynthetic. They've given up the autotrophic life to become parasites. They get their nutrients that way. But if you look in the genomes of both of those, they do have green alga chloroplast genes within their genomes. And they also have the nuclear and mitochondrial genes in their genomes from those green algae. Here's another group of protist cells that are photosynthetic primarily. These have taken up algae also, eukaryotes inside of these. These always are really odd looking because these are single cells. These aren't multicellular, but they build plates. Many of them build plates on the outside of the cells, mainly for protection. But again, these have a eukaryote, an alga, inside of the protist cell, another eukaryote, and the mixing of those genomes also. These are Rhizaria. They've also taken up green algae. That is a eukaryote within a eukaryote, and that's become their organelle. And these are mainly silicaceous cell walls on the outside. So again, these are single-celled organisms. Although they look multicellular, they are unicellular. These are stramenopiles, and includes water molds, and they're not fungi. These are protists, diatoms, and brown algae, as well as others. Instead of taking up a green alga inside of these protist cells, these contain red algae within the protist cells. So the organelle is actually from a red alga, so a eukaryote inside of a eukaryote. These down here, although they're called brown algae, they're not really algae. That's just a common name for them. So their organelles consist of a red alga, a plant of plant origin, and inside of a protist cell, which was originally not an autotroph, non-photosynthetic. So while plants have a cyanobacterium that became the chloroplast, these brown algae have a red alga, a eukaryote, that has become their photosynthetic organelle. So that originally that algal cell, the red alga, does have a cyanobacterial origin for its chloroplasts. So it's essentially the cyanobacterium inside of a protist cell that became photosynthetic, and that all of that is inside this protist cell to become the brown algae. So then there's this group, the alveolata, that includes paramecia as well as dinoflagellates and again, these are unicellular organisms. Some are quite ornate with their cell walls. Many of the dinoflagellates are a red alga inside of a protist cell, which became that, then that algal cell became the organelle. But then some of them have been further essentially swallowed up by other protist cells, and so it's a alga within a cell within another cell. Both of those are eukaryotic cells. So then you have, again, a mixing of a large number of genomes together and creating all sorts of different possibilities for what sorts of structures and proteins to build for those organisms. So to get back to this diagram, which we've talked about in some of the previous videos, when you have some simple things happening, a prokaryote, the mitochondrion inside of a eukaryotic cell, here becoming the animal cell, we have a prokaryote, an alpha proteobacterium, and a cyanobacterium in plant cells. Some of those DNAs and many of the DNAs move into the nucleus for expression, and then they're lost from the organelles. And then in these dinoflagellate cells and other, the stramenopiles, the excavata, the microbians, etc., you have a eukaryotic cell within a eukaryotic cell, and then you also get movements of all these genomes, mainly moving into the main nucleus of the cell. Now, in some dinoflagellates, then there's a further endosymbiotic event forming even more complex organelles, where there's another surrounding eukaryotic cell surrounding this. And again, most of the DNA winds up in the main nucleus of those dinoflagellates. Some of them have very, very large genomes, as you can imagine, by essentially gobbling up other organisms and using those for your organelles. Okay, so we've talked about some of these endosymbionts that become organelles, but I just want to mention a few of the other organelles, membrane-bound organelles in cells, including lysosomes in animal cells, which have degradative enzymes. So if a substance is taken into a vesicle, into the cell, often it's then fused with a lysosome to digest the contents of that vesicle. And those all have single membranes and appear to be just part of the membrane system of that large cell. There are also, of course, the nuclear membrane, which is a single membrane really folded over on itself, and that's continuous with the endoplasmic reticulum, and in particular the rough endoplasmic reticulum, which is where protein synthesis occurs for those proteins that are either going to be inside a vesicle, excreted from the cell, or wind up in a membrane. Those are continuous with the smooth endoplasmic reticulum, where other types of compounds are produced, including some lipids and membrane materials, etc. Some small vesicles then bleb off from the endoplasmic reticula. Those are transferred to the Golgi, and the Golgi often is for altering or modifying some of the proteins and other contents within those vesicles, such as adding side groups to them, sugar groups, etc., And often then vesicles from the Golgi are fused with the plasma membrane of the cell, and then those are excreted or joined with the main membrane, the plasma membrane of that cell. In plant cells, you also have similar membrane-bound structures, including, again, the nucleus, which is a single membrane system folded over on itself to look like a double membrane system, the rough endoplasmic reticulum and smooth endoplasmic reticulum, Golgi, as well as lysosomes. And then we've already talked about double membrane organelles, including chloroplasts and mitochondrion. And then there are peroxisomes also within plant cells. These are responsible for modifying products from both the chloroplasts and the mitochondria, and often they're oppressed to one another. So you'll find a chloroplast, which is right next to a peroxisome, and that's right next to a mitochondrion. And the reason for that is they're modifying products from both of those organelles and passing them back and forth to complete some reactions, including some of the reactions of the Calvin cycle. And most plant cells also have vacuoles, or one large vacuole. That is both to maintain turgor pressure, that is, because the cell wall is stiff, cells need some way to maintain an equal size and equal pressure given the constraints of the cell wall. But they also have different reactions that go on inside of the vacuole, and so it's normally acidified. And in some plants, parts of the Calvin cycle are done inside the vacuole, and other parts are done in the chloroplast. So it has multiple functions in a cell. It's not just a garbage can or just to control the turgor pressure, but it's a more complex organelle than that. Some bacteria also have internal membrane-bound structures, mainly photosynthetic bacteria, so they will invaginate parts of their membrane and form these small vesicles, and those are necessary in photosynthesis. They have actually an electron transport chain, and they also concentrate the photosynthetic apparatus within those balls. And some of the photosynthetic bacteria form spheres, spherical vesicles, and others form more lamellar or layer-like inclusions within their cells too. So bacteria, contrary to popular belief, do have membrane-bound structures within them. Some bacteria anyway, not all of them. So on the bottom here are artist depictions of those, and up in the upper right corner you can see two types of bacteria, one on the left having the spherical balls inside of it for the photosynthesis process, and these ones having the layered vesicles with inside of them containing the photosynthetic apparatus. So you would find these in some of the proteobacteria, chlorobi, acidobacteria, chloroflexus, in general, different photosynthetic bacteria. And then we come to this group of bacteria, the planktomycetes, which I've mentioned in other videos. They do have membrane-bound structures, and this is one in Gemata obscurula globus, which has a double membrane structure with the DNA inside and some ribosomes inside. So it looks sort of like a nuclear envelope, and it looks very much nucleus-like. This may be where nuclei came from in eukaryotes. There also are some electron micrographs that appear to show protein structures running through those two membranes that look quite a bit like nuclear pores. So this may have been the source of nuclei in eukaryotes. There are several groups of planktomycetes with different membranes. So this is Gemata over here, which appears to have a nuclear envelope, that is the nuclear membrane, and the DNA is housed in there, so it looks quite a bit like a nucleus. And then there's a riboplasm or a periplasm around that. And then the cytosol, the cytoplasmic part of the cell here in red. Other groups of planktomycetes have other arrangements. So they have a cell wall, and then inside of that is a cytoplasmic membrane, which is clear. And then inside of that is like a cytosol or cytoplasm inside of that. And then they have membranes that enclose part of different compartments within the cell. And so the cell is compartmentalized by these membrane systems, which is unusual for bacteria, but they really have separated organelles. One of the reasons for separation in this case down here is they have an anamoxisome. It undergoes, this is where the reactions for anamox nitrogen metabolism occur, and that's where ammonia is concentrated. The ammonia, if it were just in the cell, would start to destroy some of the proteins, the DNA, the RNA. So the reason this is separated in a compartment, separated from the rest of the cell, is to protect the other parts of the cell, but still be able to do that part of nitrogen metabolism, which is important to this organism. So again, I wanted you to see these structures in planktomycetes. They are membrane bound, and they are essentially organelles, compartments in the cell that do specialized functions for that cell. So in many cases, they look sort of like on their way to becoming a eukaryote or something like a eukaryote rather than a bacterium. So that concludes this video. In a subsequent video, I'll talk about how proteins make it into these different locations in a cell. So not only for bacteria, but especially for eukaryotes with a bunch of different compartments. How do the proteins get to different places? So we'll talk about protein trafficking within a cell. Okay, so that's it for today. For this video, I will talk to you later.